Hello. So, uh, Swechha, you are having any doubt in the previous class or whatever sir has taught or whatever I have taught or in any kind of assignment?
Not yet, ma'am.
Okay, okay. So, today I'll be teaching about the complement system, and here you can see this green color. These are the complement proteins. As you know by the name itself, complement means like it is complementing or helping in the function of some other proteins. So complement are a group of serum proteins, 50 plus serum proteins that cooperates with both innate as well as adaptive immune system to eliminate pathogens, dying cells and immune complexes from the body. So uh, it is like helping the innate as well as adaptive immune system along with our other components like antibodies or the neutrophils, macrophages, etc. to destroy the pathogen as well as other immune complexes uh, from the body. So it was first discovered because of its ability to complex with antibodies and destroy the membranes of antibody bound cells. So whenever there is an antigen antibody interaction, the complement proteins can bind with the antibodies and can form a complex which will destroy the cells which are bound to the antibody. So first it began, the research in complement began in 1890 by Jules Bordet when uh, showed that sheep antiserum to the bacterium Vibrio cholerae caused bacterial lysis or membrane destruction, and when that antiserum was heated, it destroyed its bacteriolytic activity. So, uh, what he did was he took some antiserum from sheep and first the sheep was infected with Vibrio cholerae. It is a bacteria which is calling cholera, and then like antiserum was taken from that sheep, infected sheep, and it was seen that it is causing bacterial lysis. And when that antiserum was heated, it destroyed the bacteriolytic activity of that antiserum. So when fresh serum was added to that heated serum, they again could uh, lyse the bacteria. So, however, that fresh serum was not from a sheep infected with the Vibrio cholerae. It was a fresh serum from a healthy individual. So, it was proposed by Bod at that time that bacteriolysis required two substances. One is the heat stable cholera specific antibodies that bound to the bacterial surface, and the second heat labile or heat sensitive component responsible for the lytic activity. So we all know that antibodies will be heat stable kind of. So the antibodies will go and bind to the antigens that antigen of the Vibrio cholerae bacteria, and that will bind to the cells of the bacteria. But apart from that, whenever he was heating that antisera, it is losing its lytic property. That means some other components is also necessary for the lytic activity. So what was done was this second non-specific component was purified by him, uh, and he uh, develop some antibodies specific for RBC and use them in combination with purified fraction from serum to identify which are the serum proteins that cooperated with antibodies to induce hemolysis, that is, lysis of the red blood cells. So, apart from him, Paul Ehrlich also conducted similar experiment, and they found out that there are proteins present in serum which is actually complementing the activity of the antibodies, and thus they gave it as a name as complement, and which means that the activity of blood serum that completes the action of the antibody. So there are different types of uh, complement proteins which are present, and there are different pathways of complement system. So first is like the complement pathways are initiated by proteins that bind to pathogens. Either they can directly bind or they can bind via an antibody which is binding to a antigen of that pathogen. So after there is a conformational change, here you can see these are the initiators like C1q, NBL, phycolins. Then after that, uh, there will be a conformational change by which there will be enzymatic mediators that will activate other enzymes that generate the central proteins of the complement cascade, the C3, C5 convertases, which cleave C3 and C5, releasing active components that mediate all functions of complement, including opsonization, inflammation, generation of the membrane attack complex, etc. So the effector complement proteins can label an antigen antibody complex for phagocytosis, opsonins, initiate inflammation, anaphylotoxins, or bind to a pathogen, and can help in the formation of a complex known as membrane attack complex. So they will act through complement receptors that are present on the surface, or they have some other proteins that limit the effect of uh, other like proteins which will degrade these complement components and prevent the deposition of components. So this is the overall view by which complement proteins will act. There will be initiators that will bind to either directly or to the surface of the or to the antibodies. Then there will be enzymes which will cleave the other enzymes and will activate the other proteins. Then there will be there can be different function like formation of membrane attack complex, anaphylotoxicity. There will be opsonization, and last it can lead to inflammation, and there will be complement receptors which are present on the surface of the cells through which this complement will act, and after the function is over there will be degradation of the complement components. So these are the various different types of complement proteins which are present in human system. So first initiator complement components, like they will initiate their respective complement reaction by binding to particular soluble or membrane bound molecules. Once activated by their ligand, they undergo conformational changes resulting in changes in their biological activity. Then there is enzymatic mediators. For example, there are certain components or proteolytic enzymes that cleave and activate the next member of a complement reaction sequence. And proteins that are inactive until cleaved by proteases are called zymogens. So from the inactive protein, they will cleave to form a active protein. So then there will be phagocytosis enhancing components or opsonins. So when this complement cascade is activated, there are several complement proteins which will be cleaved into two fragments, each of which then take on a particular role, and some of them will enhance phagocytosis or opsonization, and will destroy the pathogen. There will be some fragments which will bind to the receptors on the endothelial cells of blood vessels, and they will increase the capillary diameter so that there will be increase in the blood flow to the affected area, and they can also attract other cells to the site of tissue damage. And these effects, when there is a increase in the effects of this, like increase in the capillary diameter or increase in blood flow, blood pressure, it can lead to increase in blood pressure also. And then these fragments will be known as anaphylotoxins, means against protection. For example, C3A and C5. Like these proteins in excess can lead to anaphylotoxicity. So there are other proteins known as membrane attack proteins which will form a membrane attack complex, and they will insert into the cell membranes of invading microorganism and punch holes that result in the lysis of the pathogen. Then it will form a ring-shaped multiple of complement proteins with a central hole through which cytoplasmic content can escape. It can also form on infected host cells, although the complement system must overcome the regulatory mechanism designed to protect host cells from complement attack. So it is one of the last step in the complement pathway where it will form a MAC complex which will form a pore into the infected host cells. Apart from that, there are receptor proteins that will bind to these complement proteins on different types of cell surface. And for example, some complement receptors such as complement receptor 1 bind to complement proteins such as C3b that have opsonized pathogens triggering phagocytosis of the C3b bound pathogen. Another is C5a receptor which will bind to the component protein C5a on neutrophil to stimulate neutrophil degranulation and inflammation. There are also regulatory complement components like to prevent unwanted damage, complement mediated damage, they will regulate the function of these complements. For example, it includes factor 1, which degrades C3b, and CD59, which inhibits the formation of MAC on host cell. So these are the overview of what are the different components and how they will inhibit the pathogen inside the cells. So there are three major pathways of complement activation. One is the classical pathway, one is the lectin pathway, and one is the alternative pathway. So there are different steps in the complement pathway. Here, classical pathway, you can see it depends upon the antigen antibody immune complexes. However, in lectin pathway, it will recognize the lectin or these carbohydrate molecules present on the surface of the pathogen. And there in the alternative pathway, there will be spontaneous hydrolysis of these complement proteins. So depending on the types, they are named as classical pathway, lectin pathway, and alternative pathway. So initiating and amplifying the proteins of the classical and lectin mediated complement pathways. So these are the different molecules like IgM and IgG. It can bind to the pathogen surface and initiate complement cascade, and this belongs to the classical pathway. There are mannose binding lectin or phycolins which are present to which bind to carbohydrates present on the microbial surfaces and initiate complement cascade. This belongs to the lectin pathway. Then there is C1 which gets cleaved into different fragments like C1q, and they can initiate the classical pathway by binding to the immunoglobulins, or they can initiate phagocytosis or apoptosis, and they also belongs to classical pathway. Then there is MAS1, which is MBL associated serine protease 1. MAS2 appears to be functionally more relevant MAS protein. It also belongs to lectin pathway. Then there is MASP2. This is also a serine protease, and it also belongs to lectin pathway. It cleaves C4 and C2. Then this is C2, which will get changed to C2A. She is also a serine protease, and with C4B it forms a complex which is known as C3 convertase. It belongs both to classical and lectin pathway. Then there is C2B, which will mediate the cleavage by plasmin release C2 kinase, a peptide that stimulates vasodilation. And there is C4. It also contains various fragments like C4b, C4c, C4d. So C4b I told along with C2, it will form C3 convertase and it will convert C3 into different fragments. It also belongs to classical and lectin pathways. Then there is C4c and C4d that will lead to the cleavage of the products generated by one factor 1, which is I told as already, it is a regulatory complementary component. Then there is C3, which will be cleaved into different fragments, C3a, C3b, IC3b, C3f. So the C3a will interact with the C3 alpha receptor. It will lead to anaphylotoxins, and it also belongs to classical lectin pathway. Then C3b will help in opsonization. It can tag the immune complexes, pathogens, apoptotic cells for phagocytosis, and with C4b and C2a, it forms again a complex which is the C3 C5 convertase. Then there are other proteins like IC3b and C3F, which will be generated by factor IC3b, which binds to receptor CR3 CR4, and it also will it is just a proteolytic fragment of C3b. Then C3b, C3d, and C3dg are also proteolytic fragments of IC3b generated by different proteases, and they also facilitate antigen binding to B cells. Then there is C3c, which is also a proteolytic fragment of IC3b, and it binds to C-RIG on fixed tissue macrophages. So these are the different types of molecules which are present mostly in the classical as well as lectin complement pathways. Then there is another pathway, alternative pathway, where also similarly different types of factors are present. For example, similar to C3, what I told, here also all the components of C3 are present. Then apart from the C3, there is the presence of a factor B. This factor B, what it does, it gets binds to C3, hydroxylated C3, and is then cleaved by factor D into two fragments, BA and BB. So BA is a smaller fragment of factor D mediated cleavage of factor B, and it can inhibit the proliferation of activated B cells. So BB is a larger fragment of factor D mediated cleavage of factor B, and it can access fluid phase C3 convertase, and with C3b it can access cell bound C3 convertase, and with two molecules of C3b it can access C5 convertase. Then there is factor B, which is a proteolytic enzyme that cleaves factor B into B and BB only when it is bound to either C3OH2 or to C3b. This factor B is cleaved by this factor D. Then apart from that, there is propardine. This propardine stabilizes the C3b8b complex on microbial cell surface. So these are the different components of alternative pathway. Then there is a component of the membrane attack complex, which is common to all the three pathways, that is the classical pathway, the lectin pathway, and the alternative pathway. So these are the molecules which is forming this membrane attack complex. So there is C5, C which is getting cleaved into C5 and C5b. C5a is an anaphylatoxin which is binding to C5a receptor which induces inflammation. Then there is C5b which is also a component of membrane attack complex. It binds to cell membrane and facilitates binding of other components of the MAP. Then there is C6. C6 is also another component. It stabilizes C5b, and in the absence of C6, the C5b cannot be a part of the MAC and it is rapidly degraded. So it is stabilizing the C5b. Then C7, it binds to C5b C6 complex and induces conformational changes, allowing C7 to insert into the interior of the membrane. So all these are forming a complex C5, C6, C7, C8 and C9. So then the C8 will bind to the C5b C6, C7 complex and it will create a small pore in the membrane. Then the C9 will bind to the C5b C6, C7, C8 and create a large pore in the membrane. So this is forming a big complex which is forming a ring-shaped complex which will bind to the membrane and they will create a pore in the membrane. So this is the classical pathway which is initiated by antigen and antibody binding. So here you can see this is the C1 complex. Here is the antigen. Here is the antibody binding to this antigen IgG antibody, and this whole C1 complex is binding to the Fc region of this antibody. So here C1q interacts with two molecules of its C1R and C1S to create the C1 complex. The C1q molecule consists of 18 polypeptide chains of in 6 collagen-like triple helixes. So this is a big complex which is forming like C1q, C1R, C1S. So that whole together they are forming the C1 complex and it is interacting with the antibody which is interacting with the antigen on the surface of the pathogen. So what happens? Here C1q binds to the antigen bound antibody. Here this is the antibody, this is the Fc region. The C1q is interacting with the Fc region of the antibody. Then and it induces a change in the C1R molecule. Here C1R molecule and it is activating it. This C1R can then activate the second C1R and two C1S molecule. So here C1q is binding which is activating C1R which is getting activated, then C1R then activates a second C1R molecule and two CIS molecule. After this C1S will cleave C4b and C2. C4 and C2. I already told that the C2 and C4 will form a complex which is C3 convertase. So the C4 is cleaved into C4b which is binding to the membrane close to C1 and C4b binds to C2. Here you can see a complex C4b and C2 is binding and is forming a complex which is like after binding it exposes it to the action of C1S. Then C1S can again cleave. So C1S is basically a protease. Here you can see C1S is a serine protease cleaving C4 and C2. So C1S is a protease which is a cleaving first C4 and then it is cleaving C2 and then after cleaving C2 it can create the C4 convertase which is the C4b, C2a. The C2b is going, it is not taking any part, but C4b and C2a is combining into C3 convertase. The C3 convertase we can hydrolyze many C3 molecules, some combine with C3 convertase to form C5 convertase. Here C4b2a is there, which is C3 convertase, and then it can hydrolyze many C3 molecules. Here you can see C3 molecules, and it will form a complex, like it will also form C3b, C3a, and the C3b will form a complex with the C4b and C2a and will form the C5 convertase. The C3b components of the C5 convertase bind C5, permitting C4b 2a to cleave C5. So this C3 also is this is a serine protease, as I told. It is C3 convertase, which is converting it into C3a, C3b, which is forming a complex, which is a C5 complex, which will then bind to this C5 and it will cleave C5 into C5a and C5b. So all the proteins are cleaving one another to form a complex-like structure. So C5, as I already told, C5 is a component of the membrane attack complex. Any kind of doubt is there, you can ask in the message box, or you can unmute yourself also.
No, ma'am.
Okay. So this C4b here can bind to the microbial membrane. It can bind through, here you can see that C4b is binding directly to the membrane before binding to C2, and it can bind to the microbial membrane surface through a thioester bond via an exposed amino or hydroxyl group. So these both C3b and C4b contain highly reactive thioester bonds which are subject to nucleophilic attack by hydroxyl or amino groups on cell membrane proteins and carbohydrates. So this is unbound C4b. Here it is, cell membrane is there, and then this is a thioester bond. Then in the cell membrane, you can see when it is unbound, there is no the bond is intact. However, this bond will break whenever it wants to bind to the membrane. So there will be a nucleophilic attack by the hydroxyl or the amino group on the cell membrane proteins and the carbohydrates, and this thioester bond will break, leading to the covalent bond formation between this membrane macromolecules and the complement components. And if this is not happening, if this covalent formation does not occur quickly after generation of the C3b and C4b fragments, then the thioester bonds will be hydrolyzed as shown. Here you can see it will be hydrolyzed. These thioester bonds will be hydrolyzed. So this is just a mechanism through which this C4b will bind to this membrane. So one of the important uh, part of this complement system is the generation of C3b. How C3b was forming? C3b was forming via this serine protease C4b2a, which is cleaving this C3 to C3b and C3a. So it has been seen that deficiency of complement proteins that can act prior to the C3 cleavage can leave the host extremely vulnerable to both infectious and autoimmune diseases. And however, if patients are deficient in C3, they are susceptible to infection with both gram negative as well as gram positive bacteria. Because why? Because these complement proteins are actually helping the antigen antibody or the immune complexes or the other through other pathway also. It is helping it to remove the pathogens from the body. So if there is no complement proteins, as you or when you saw that when there was like they heated the serum anti-serum and the complements were destroyed, there was no antibacterial or antibacteriolytic activity of the Vibrio cholerae. So you can understand like how much important these complement proteins are, and if anyone is deficient in this complement system, they are susceptible to all kinds of infection, or they can suffer from any autoimmune disease also. So C3b is very important, and there are why there are three important and different ways by which C3b can protect the host. Like C3b, C4b, C3b can also bind to this microbial surface by same mechanism, and they can provide a molecular tag that allows phagocytic cells with C3b receptors to engulf the tagged microbes. This process is called opsonization. So there will be C43b receptors present on the surface of the microbes, and this C3b can interact with these receptors, and they will tag that this is a microbe and we should destroy it, and they will help in the process opsonization. So C3b can also attach to the Fc region of the antibodies participating in soluble antigen antibody complexes. So these C3b tagged immune complexes are bound by C3b receptors on phagocytes or red blood cells and other phagocytosis or conveyed to the liver where they are destroyed. So they can also help in the process of phagocytosis. Another is some molecules of the C3b can bind the membrane localized C4b2A enzyme to form the trimolecular membrane-bound C5 convertase complex C4b2A3b. So they can either opsonize or they can either phagocytosize, or they can also form a complex like with the C4b2 enzyme C5 convertase complex, which is important for the generation of that membrane attack complex. So these are the three important functions of C3, and why this C3 deficiency is causing host susceptible to infectious autoimmune disease is because this is helping in the functioning of this many processes. So this is the classical pathway. Then comes the lectin pathway. So, as I already told, classical pathway is dependent on antigen-antibody complexes, while lectin pathway of complement activation is dependent on the uh, recognition of uh, lectin molecules or carbohydrate molecules uh, on the surface of the antigen. So, instead of pathway also, like the complement pathway, can proceed through the activation of C3 convertase composed of C4b and C2m. So instead of relying on antibodies and antigen complex to recognize a microbial threat and to initiate the complement activation process, this pathway uses lectins, which are proteins that recognize particular carbohydrate components as its specific receptor molecules. So for example, one is some mannose binding lactin or members of the picoline family, which can recognize the microbial surface carbohydrates, and these carbohydrates will bind to these receptors and this pathway will be activated. The lectin provides the recognition function, binding to carbohydrate residues on a microbial surface. This binding activates an associated serine protease MASP2. Here you can see MSP1, MASP2 that cleaves C4 and C2 to create the C3 convertase C2A, C4B. So there we saw that classical pathway is getting started via the C1q molecule. But here we are seeing that these are there are different types of receptors which can recognize the carbohydrates of the lectin molecules on the surface of this uh, pathogen, and they are getting initiated by the. There we saw C1s as a serine protease, which is cleaving C4 and C2. Here there are other serine proteases, which is MAS1 or MAS2, that is cleaving this C4 and C2 to create the C3 convertase C4a to B. And as for the classical pathway, the end result of the initiating sequence of the lectin pathway is a generation of enzymes that cleave C3 into C3a, CB, C3b, and C5 into CA and CB. So everything is similar except the like the binding of the complement molecules to the surface antigen. Like there it was like antigen antibody complexes with which that C1q molecule was binding. Here it is the presence of this carbohydrates on the surface of this uh, microbe to which these specific receptors are binding, and after that the pathway is similar, like this instead of C1s there is MAS1 or MAS2, which is actually cleaving this C4 and C2 and thus creating the C2A4B, which is a C3 convertase, and then the C3 again is getting cleaved by the C3 convertase into C3A and C3B, and then they are forming a complex, and then it is also cleaving C5 into C5A and C5B. So this is the ultimately it can also lead to the formation of the membrane attack complex. So these are the lectin pathway and the classical pathway. Then there is another pathway which is the alternative pathway, and it is initiated in three different types. There are three type of pathway. Like one is the takeover pathway, alternative takeover pathway. Takeover means that C3 is constantly being made and spontaneously inactivated and is thus considered to be taking over. Here you can see this is taking over of the C3. C3 is constantly made, then it is forming complex with factor B, then it is hydrolyzed, then factor D, then it is cleaving that B and BB, then this whole complex will form a C3 convertase. So this is like why this is called taking over, because it is constantly made and destroyed, constantly made and destroyed. So it is a taking over pathway, alternate pathway. Then spontaneous hydrolysis of the soluble C3 to C3H2O allows the altered conformation of C3H2O to bind factor B, which makes it susceptible to cleave by factor D. The resulting complex C3H2O-BB forms a fluid phase convertase capable of cleaving C3 to C3 and C3B. Then some of the C3B molecules here formed by the fluid phase convertase binds to cell membranes. Here you can see C3 convertase can bind to the cell membrane via that thioester formation, and C3B like C3H2O can bind to factor B, again will cleave uh, this factor B to BA and B, and it will form. Another is membrane bound C3B stabilized by another factor, which is factor P or properdin, which binds to the C3BB complex on the membrane, and addition of another C3B to the C3BB complex forms a C5 convertase, which is also stabilized by properdin. So these are the different mechanisms by which this alternate pathway is activated, and they also will form this C3 complex, C3 convertase complex. There will be two types like fluid phase and membrane bound C3 convertase. Fluid phase means in the fluid it can act, and so it is hydrolyzed. But when there is a need for membrane boundation, which is hydrophobic, as you all know, membrane are hydrophobic, so it need not be hydrolyzed, and so it is forming in a different manner. Another is that it sometimes gets stabilized by another factor P, which can again lead to the formation of the C5A, C5B, which will can form the MAC complex. As I already told, this properdin or factor P is stabilizing this C3B and complexes, and uh, it then gets cleaved, then forming complex with factor B, and then which is then cleaved by factor D into C3B PBB. So this is just different in the fact that properdin first bind to the uh, like first properdin binds and stabilizes the C3 rather than other factor B or factor D, and then initiate the complement deposition on the membrane. So these are the three different pathways: classical pathway, alternate pathway, and the lectin pathway, which are getting activated whenever there is a invasion by any kind of pathogen. So any kind of doubt is there? Okay. So as I already told, these three complement pathways will converge and the formation of C5 convertase, because in each uh, pathway you can see this C5 convertase is forming. Here also C5 convertase will be formed. Also C5 convertase will be formed, which will result in the formation of this MAC complex. So here we can see C5 into C5B and C5A, where it is forming complex with C6, C7, and C8. Here you can see C5, C7 is getting inserted into the membrane of the cell, and then C8, then C9, and all this is forming a ring-like structure, a poly-C9 complex. I told, like many C9s are binding, 10 to 19 C9s are binding together and is forming a ring-like structure, and it can induce lesion on the membrane of a red blood cell. Here you can see it is forming pore. You can see this is a red blood cell, this is the electron microflora, and here you can see lesions by the forming by the membrane at a complexes. Then this is just a structural, and here C9 is forming the ring, and then it is forming a pore on the surface of the membrane. I am coming just in 5 minutes. So there are different types of functions which are uh, there uh, of complement. It is having different types of functions. Like first is there is innate defense against infection. There is lysis of bacteria as well as cell membrane by the membrane attack complex, that is the complex by C5b, 6, 7, 8, 9. There is opsonization by covalently bound C3b, C4b. There is induction of inflammation and chemotaxis by anaphylotoxins, C3a and C5a, and their receptors on the leukocytes. There is then interface between innate and adaptive immunity also. I told it is helping in the antibody responses like C3b and C4b. C3b and C4b is actually uh, binding to these immune complexes and antigen, and also there are C3 receptors present on the immune cells. Then there is enhancement of immunological memory, for example C3b and C4b, and their fragments are binding to antigen and immune complexes and receptors for complement components on follicular dendritic cells. Then there is enhancement of antigen presentation by this minus binding lectin C1q, C3b, C4b, and C5a, and it can also have effect on the T cells by C3, C3a, C3b, C5a. And in the contraction phase of the immune response also, it is helping. Contraction means when the immune response needs to be stopped, that it should not be like overactivated. That phase also these complement proteins are helping, like they are helping in clearance of the immune complexes from the tissues, for example C1, C2, C4, and covalently bound fragments of C3 and C4. Then there is clearance of apoptotic cells, then like C1q, and covalently bound fragments of C3 and C4. Loss of CD46 triggers immune clearance. Then induction of regulatory T cells by CD46. So these are the different kinds of functions of complement proteins. And here, these are the different types of complement receptor. As I told, these complement receptors are bound to many types of cells. For example, CR1, CR2, CR3, CR4, C3AR, C5AR, C5L2. So these are mostly present on the immune cells and as well as red blood cells, because it is held, as I already showed you, this graph, this electron micrograph, where it is showing like they are making a hole or the pore on the surface of these red blood cells. So all these receptors are present in mostly monocyte, macrophage, eosinophil, then erythrocytes, and all these cells, and they have different types of functions like CR1, or it has other name also, CD35. These are the names. As I already told, CD means cluster of differentiation. These are different CD molecules are present on the surface and mostly acts as co-receptors. So this CR1 helps in the clearance of immune complexes, enhancement of phagocytosis, regulation of C3 breakdown. CR2 in also B cell activation, B cell co-receptor, retention of C3D tagged immune complexes. CR3 in binding to adhesion molecules on leukocytes facilitates extravasation. IC3B binding enhances opsonization of immune complexes. CR4 is IC3B mediated phagocytosis of bacteria by MZ macrophages. The C3A receptor is induces degranulation. In C5AR also it acts as a chemoattractant. It induces degranulation. It acts with IL-1 beta and TNF alpha to induce acute phase response and induces respiratory first in neutrophils. Then there is C5L2, which is uncertain, but most probably it can downregulate pro-inflammatory effects of C5A. So these are the different functions of the complement receptors present on the surface of different kinds of cells. So here is an example, just like B cell co-receptor is a complex of three membrane molecules, CD21, which is also known as complement receptor 2, CD81, TAPA1, and CD19. These are the three co-receptors present, co-receptor complex present, which is helping in the B cell recognition and activation. So antigen here is bound to the antibody, IgM antibody, and here you can see the CR2 or the complement receptor 2 is bound by C3d fragment, and this C3d fragment is bound to both this receptor as well as the antigen, and thus it increases the avidity of the cell receptors for the antigen, allowing only lower concentration of antigen to activate the B cell. So this is helping in to bridge the gap between this antigen as well as the uh, like the activation of the B cell. Like, so even small amount of antigen which is recognized by this antibody as well as this complement receptors can be able to trigger B cell activation. So here you can see these, as I already told, there are C3 and C5 are known as anaphylotoxin because in excess they will be harmful to the cell. So these anaphylotoxins will bound to a G protein coupled receptor, C3AR or C5AR, and they will stimulate the release of many pro-inflammatory mediators from macrophage, neutrophil, basophil, eosinophil, and mast cells, and which will actually help in either if in normal situation if there is a pathogen, it will help in clearance of that pathogen, but if in excess they can lead to uh, any kind of toxicity or any kind of stress to the cell which is not good for our system. So they are known as anaphylotoxins. Here you can see they can activate the macrophage, basophil, eosinophil, neutrophil mast cell, and they can cause degranulation, releasing enzymes. There can be oxidative burst. There can be phagocytosis. Anything can happen by this anaphylotoxin signaling. So there are functions, as I've already told. There are different functions like it can directly lyse the cells. It can act as a it can reduce the gap between innate and adaptive immune response, and it can trigger or activate T cells, B cells, etc. So there are different functions also, and it enhances the host defense against infection. So, as I already told, MAC or membrane attack complex, it can induce cell death via cytolysis. For example, the foreign microorganism like viruses, fungi, and damaged erythrocytes can be lysed by this MAC, and the cytolytic mechanism is considered to be penetration of the cell membrane leading to uncontrolled influx of ions and water down to osmotic gradient, causing it to swell and ultimately burst. So if there is a pore in the membrane, so what happens? From the outside, many things can enter into the cell, and there will be a change in the osmolarity of the cell, and it will cause the cell to swell. If more and more water is entering, then it will swell the cell, and it will burst. There is also production of cytokine, like whenever there is a insertion of this MAC complex, it can increase the membrane permeability, and many its pathways will be triggered to produce various bioactive substances like these inflammatory mediators like oxygen free radicals and cytokines, and these have a positive effect on pathogen clearance and amplification of inflammatory reaction. Then there will be adhesion molecule expression. For example, they will promote the expression of these adhesion molecules, which can increase the adhesion of these neutrophils, platelets, and other cells that will accelerate the release of inflammatory mediators. Then there will be sublethal effects like recent studies suggest that karyocyte can remove MACs deposited on their cell membrane through vesiculation or endocytosis to protect themselves from complement dissection or dissolution. So these are the different kinds of function by this membrane attack complex, and this is the ultimate, like the membrane attack complex, as I told already, is are common to all the three pathways, and it will help in clearing the uh, like the infection. So here is the MAC is almost effective against gram-negative bacteria, nucleated cells, and envelop viruses. So this is the complex. This figure I already have shown you. Here poly C9 complex, which is forming the pore and clearing the pathogen or lysing the cell. So as I told already that it also promotes opsonization. So what is opsonization? I have taught in the last class. Like opsonization is the capacity of antibodies and complement components as well as other proteins to coat dangerous antigens that can then be recognized by the Fc receptors present on the antibodies or complement receptors on phagocytic cells. So it is an extra added effect. Like when it is bound by the uh, complement proteins, these antigens are bound by the complement proteins, it will enhance the activity of the antibodies or other complement receptors, which will then recognize and opsonize the cells and will ultimately phagocytose. So it is phagocytosis are also mediated by different kinds of complement receptors present on the surface of the macrophage and the neutrophils, like for example CR1, SINE R1, C1QRP. Then also this antibody has this Fc region which will bind to these Fc receptors, can also bind to antigen opsonized by antibody binding. So these complements also help in the uh, recognition as well as opsonization and phagocytosis of this uh, pathogens. So as I already told, it can act as an uh, interface between innate as well as adaptive immunity. For example, in case of antigen presenting cells like dendritic cells and follicular dendritic cells, macrophages, they express many of these complement receptors. So when these receptors are bound to the antigens, uh, these complement proteins can actually help in the uh, signaling to their respective receptors to enhance antigen uptake. And this signaling of these antigen presenting cells through this uh, C5AR, for example, which is a receptor for C5A, is shown to modulate the migration of them to affect the production of interleukin, particularly cytokine IL-12. So when there is a production of cytokine IL-12, then it will skew the T cell response towards Th1 phenotype. So it is starting from this innate immune response, and it is helping in the production or the activation of this adaptive immune response. Then there is complement and B cell mediated humoral immunity. For example, it showed that when mice was depleted of C3 complement protein, it impaired their T cell dependent antigen specific B cell responses, thus implying the complement may participate in the initiation of B cell response. It, however, now appears that this observation was the first description of the complement receptor CD21 acting as a co-receptor in antigen recognition. So, another is the complement and T cell mediated immunity. Like the immature T cells are protected from natural antibody and complement mediated lysis by the provision of additional sialic acid residue present on the surface glycoprotein. So, uh, in case of defective T cells, where they do not have this layer, this sialic acid residue on their surface, they also complement participates in quality control mechanism during their development also. So binding of the complement proteins to their respective receptors on mature T cells will facilitate their growth, differentiation and survival. So it is thus helping in to reduce the gap or the breach between adaptive as well as innate immune system. Also, as I already told, like during when there is no need of the activation of the immune system and that immune complexes has to be degraded or it has to be removed from the system, this complement also helps in that removal of these complexes or degradation of these complexes. For example, during the contraction phase of the immune system, immune response, excess lymphocytes that were the product of this antigen induced expansion are eliminated by apoptosis. The complement component C1q, which if you remember, it is a component of the classical pathway. It will recognize the presence of cell surface DNA and proteins on the outer membrane of apoptotic cells and bodies and mediate phagocytosis either directly via binding with the receptor or indirectly via after initiation of the complement pathway. So whenever there is an excess production of this lymphocyte, T cell, T lymphocyte, B lymphocyte, which are produced when it got clonally expanded, they are constantly removed by apoptosis, which is mediated by this C1q of the classical pathway. Then the antigen antibody complexes during a course of immune response are opsonized by C3b and removed from the circulation after recognition by CR1 receptors on erythrocytes. So you can see how they will help in the contraction phase of the immune response and will clear of this excess things or excess molecules which are produced in response to activation of our immune system. So here you can see one example that uh, this circulating immune complexes is cleared off by binding to the erythrocyte complement receptors and subsequent stripping from these receptors by macrophage complement receptors in the liver and spleen. Here this is a soluble immune complex antigen. This is the antigen and this is bound to the antibody in blood and there will be like binding of this and there will be binding of this complements to this complex and then this complex will then go and bind to the receptor CR1 receptor or other receptor present on the surface of the erythrocytes and then these erythrocytes will just help it to get transported to the liver and the spleen because spleen is the graveyard of RBC, so it will just go into the spleen where there will be phagocytes and these phagocytes will then phagocytose this whole immune complex and thus it will be destroyed. So uh, However, it has been seen if there is a deposition of these immune complexes in the liver and spleen or any kind of organs, there can be damage to that special organ. For example, here they have mentioned that deficiency in this process can lead to renal damage due to accumulation of immune complex. So, whenever the work is done, these complexes need to be removed from our body. So, any kind of question is there? Okay, so if anyone is having any doubt, they can comment in the message box or they can unmute and tell. So, so these complement proteins are regulated, like these are mediating the removal of these immune complexes. But what actually is removing this, like complements, because complement also can activate the other pathways and it also can lead to the activation of the immune system. Then it should also be removed from the cells. So there are different mechanisms through which these complements can be uh, degraded or destroyed. For example, there are dissociation of the C1 components. Here you can see C1 components is made of C1 R2S2. So the protein half-life of these complements are very much less. So they cannot stay longer in the system. So whenever their work is finished, they are prone to get degraded. Then the accelerating activity for C3 convertases will be decayed. There are certain molecules which will actually dissociate this uh, like complexes also. Like for example, C4b2a, which is a C3 convertase, can be dissociated by this DAP or CR1 or these molecules. Then there is one factor I, which I already told. They can also cleave this C3b into C3c, IC3b, C3dg, and also C4b into C4c, C4d. Then there will be inhibition of lysis. For example, here you are seeing the formation of this membrane attack complex, is C5b, C6, C7, C8. So this molecule protectin will act as a, you can see, umbrella or shielding, and it will prevent C9 binding and polymerization so that there will be no formation of the membrane attack complex. Then there will be cleavage of the anaphylatoxins like C3 and C5a by different carboxypeptidase N, B, and R, where it cannot uh, like make a complex with the other complement proteins. So this with the immune complexes also, this complement proteins also needs to be degraded, and these are the different pathways or the different mechanisms by which these complement proteins are getting degraded. So here also you can see uh, example they have given in this table, like C1 inhibitor is there here, which is induces the dissociation and inhibition. Then this, as I told, this DAF or the CD55, which is membrane bound and it accelerates dissociation of the C3 convertase. Then there are receptors like CR1, which blocks formation and formation of an acylation dissociation of the C3 convertase. And there is factor I, which is serine proteases, which is cleaving the C4b and C3b using the cofactors. These are the different cofactors. And there is factor H, which is actually blocking the formation, acceleration, dissociation of the C3b, BB, C3 like the convertase. Like this, others are there like S protein, this protectin, as I told, it is preventing this MAC complex, then this carboxypeptidase. So these are the regulated, these are the ways by which these complement proteins are regulated. So there are like these complement proteins, as you can see, is very much important in regulating the adaptive and innate immune response, as well as it can also like helping the antigen antibody complex to enhance the reaction, and can also degrade directly the it is doing the lytic activity also. So it can be a good therapeutic target, obviously. So first is like the involvement of complement-derived anaphylotoxins in inflammation makes complements an interesting therapeutic target in the treatment of inflammatory diseases such as arthritis. However, that there are certain autoimmune diseases like multiple sclerosis and other different macular degeneration, and these autoimmune diseases actually can result in the complement-mediated damage to host cells. If these complements are not removed from the system after its function is over, it can lead to different kinds of diseases like, as I told, multiple sclerosis and macular degeneration. They can also act as a potential candidate for complement-focused therapeutic intervention. For example, one disease is there, paroxysmal nocturnal hemoglobinuria, which is like it enhance the fragility of erythrocytes, leading to chronic hemolytic anemia, pancytopenia, and venous thrombosis. So this is a condition where there will be heavy loss of blood cells of all types. There will be formation of blood clot, and there will be anemia also. So its name is derived from the presence of hemoglobin in the urine, and most commonly observed in the first urine passed after a night's sleep. So after the sleep, if there is a presence of hemoglobin or blood you can see in the urine, this is like this condition is, you can understand that that person is suffering from this condition. So the condition is caused by a defect in the synthesis of cell surface proteins, which affects the expression of two regulator of complement, that is DAF. Here I showed already, DAF is there, CD55, and this protectin CD59. So these two are deficient in a person, and it can cause a disease known as PNH, or paroxysmal nocturnal hemoglobinuria, where there is presence of hemoglobin in the urine. So in 2004, a breakthrough occurred in the treatment of PNH, where a humanized monoclonal antibody that targets the complement component C5 was used. It inhibited the terminal steps of the complement cascade and formation of the membrane attack complex. This antibody was known as Soliris or eculizumab, was infused into patients and then monitored for the loss of red blood cells. So it was seen that after during a 12-week period of treatment with this monoclonal antibody, this relieves this hemoglobinuria. This reverses the kidney damage also, resulting from high level of protein in the urine, and significantly reduces the frequency of thrombosis also. So in 2007, it was approved as a treatment for PNH in the general population. So they made a monoclonal antibody that is targeting this component C5 in the last stage, and thus preventing the formation of the membrane attack complex. So if the membrane attack complex is not formed, it won't form a pore and there will not be excessive loss of the ions, or there won't be excessive damage to the cells, and so there will be no loss of red blood cells as they have noticed. Because in erythrocytes also it can go and form a pore, as I have already shown you. So they saw that in a 12-week period treatment with this monoclonal antibody, there was a dramatic movement in the patients, and in 2007 it was approved as a drug against this PNH. So here you can see that before they administered this monoclonal antibody, and after they administered this antibody, they saw that there was less amount of hemoglobin found in the urine compared to the patients who were not administered this uh, antibody. So you can see how much this antibody can be, this complement can have some therapeutic potential. Either if a person has more complement proteins, then you can use an antibody to actually stop the pathway, or if some person has any anti like less complement proteins, you can also supplement that person with complement in form of tablets also. So these are the different ways which you can design the therapeutics against this complement proteins. So here is an example, like here we saw that excessive complement proteins are there, and these two uh, like regulators of complement like DAF and CD59 are not there. So more and more amount of complement is there, and more amount of damage is happening to the cells. But what if there is complement deficiency? So complement deficiency also can cause different kinds of diseases. For example, here C3 deficiency, factor 1 deficiency, or secondary C3 deficiency due to utilization can cause uh, different kinds of clinical manifestation. Recurrent pyogenic sepsis, it can cause sepsis. As I already told in the first, like C3 can, people who are deficient in C3 can actually suffer from different bacterial infection, and it can be due to the, why it can be deficient, it can be inability to generate C3b and failure to generate the membrane attack complex. Then deficiency of C1q, C1r, C1s, C4s, C2, it can cause SLE, or it is autoimmune disease, systemic lupus erythematosus, and it also can occur due to generation of failure to generate C3 convertase, and it can cause defective scavenging of apoptotic cells and immune complexes. So if there is a deficiency in C1q itself, in the first step of the complement activation pathway itself, so there will be no subsequent formation of the other like steps like C3, C5, then there will be no membrane attack complex, and then there will be more and more deposition of these immune complexes, and so there will be like inflammation and other things. So they cannot remove these complexes, they cannot remove the apoptotic cells, and there will be condition like SLE where there will be inflammation in the system. Then there is a deficiency of properdin or factor D that can in the alternative pathway, it cannot thus generate C3 convertase, and there will be failure to opsonize encapsulated bacteria when anticapsular antibody is limiting. So there can be recurrent and encapsulated bacterial sepsis. So there sepsis can occur if there is a deficiency of properdin or factor D. Then when there is a deficiency of factor H or I, it can fail to regulate C3 activation. There can be hemolytic uremic syndrome, membranoproliferative glomerulonephritis. So immune complexes cannot be here also if there is no factor I or factor H, no C3 will be activated, and there will be more and more amount of these immune complexes which are getting like deposited in the liver or spleen or wherever, and it will lead to the hemolysis, or there will be like glomerulonephritis, a kidney condition. And if there is C1 inhibitor deficiency, it can also uh, regulate the it cannot regulate C1 activation and kinase generation, which can uh, result in the uh, formation of a condition which is angioedema, which is like a condition where there will be swelling under the skin, and there will be allergy to different kinds of pollens, drugs, or other things. So another is CD59. CD59 cannot protect if there is a deficiency in CD59. They, from the endogenous C3b, it cannot protect the cell surfaces, and you can see there can be hemolysis. There will be less loss of, like, the red blood cells will be hemolyzed faster, and thus it can lead to hemolysis. So these are the different types of complement deficiency which can lead to different kinds of diseases. So as I already taught in the last class, like how the pathogens can actually escape our immune system. Here also these are some of the examples where the microbes can actually evade this complement system. So some pathogens can interfere with the first step of immunoglobulin mediated complement activation by binding to the Fc region of the antibodies. These pathogens will directly bind to the Fc region so that the complement proteins cannot bind. So the complements won't be activated. The complement proteins cannot bind, then they won't bind to the receptor, and the subsequent steps will not be activated. Some viruses also enhance the rate of clearance of antigen antibody complexes from the surface of virally infected cells. So they will enhance the rate of clearance of antigen and antibody complexes by forming some strategy. Then there are also some microbes that produce the proteases that destroy the complement proteins. Some produce proteins that bind to complement proteins, masking their site of interaction. Some can mimic or directly acquire the complement regulatory proteins in order to evade complement mediated destruction. And some can synthesize regulatory proteins by the cells that harbor them. So they can, in multiple ways, they can actually bypass this complement system. For example, here are some of the example. For example, it is like they can interfere with the antibody complement interaction. One example is they will produce a protein, the staphylococcal protein A, which will go and bind to the antibody, and they will, so that Fc region, as I already told, and that complement cannot go and bind to this uh, Fc region of the antibody. Then they can also remove this IgG molecules by staphylokinase. So they have formed the enzyme also staphylokinase. Then this binding and inactivation of complementary proteins, complement proteins. For example, Staphylococcus aureus is a protein skin which binds and inactivate the C3 convertase. Then this parasite protein C2 receptor, it disrupts the binding between C2 and C4. So if you are making a similar kind of protein which is of similar size and similar shape, it will go and bind to your particular receptor, or you can bind to any kind of another protein, and thus preventing the binding between that protein and its partner. So apart from that, in Streptococcus, you can see there is protease-mediated degradation of the complement component. For example, in case of Pseudomonas, they synthesize to enzyme elastase and alkaline phosphatase, which degrades C1q, and then C3C3b, and then SCPA and SCPB from Streptococcus degrade C5a. Then there is also microbial mimicry, means they will just mimic the proteins and they will make the similar kind of proteins. For example, Streptococcus pyogenes M protein bind to C4BP and factor H to the whole cell surface, accelerating the decay of C3 convertase bound to the bacterial surface. Variola and vaccinia virus. This is like the M protein of Streptococcus pyogenes will bind to C4BP and factor H to the cell surface, thus accelerating the decay of C3 convertases bound to the bacterial surface. Then there are two viruses like variola and vaccinia, which will express proteins that act as cofactor for factor I, which will help in degradation of C3b and C4b. So you can understand like they will mimic the proteins directly, and that will prevent either the degradation or the function of the complement proteins. So microbes are very much cleverer than us, and they are also constantly evolving to form or bypass the host system. Then there last week also I showed like how much there is evolutionary conservation among different types of uh, vertebrates as well as invertebrate in terms of adaptive or innate immune system. And as you all knew, like this innate immune system is present in all kinds of animals, be it invertebrate or vertebrate, but adaptive has not been developed in other vertebrates, or studies are still going on. So here also this is a table which shows whether which pathways are present in which organisms, and since mammals all the pathways are present like alternate pathway, classical pathway, lectin pathway, membrane attack complex, and antibodies. Here you can see in birds also all are present. In reptiles also all are present. Till fish everything is not till fish everything is present. Then this Agnatha fish, here still it is not known whether antibodies are present or not, and this classical pathway is not present. Then other like echinoderms, tunicates also, these are not very much uh, like developed. So complement system is mostly present in almost all the mammals and birds and reptiles and amphibians, and not all the fish but fish. So you can understand how they have like evolutionary, they are conserved among all the like animals, and maybe their proteins are somewhat different if you check, but more or less this complement system is very much evolutionary conserved, and you can understand how much that means they are playing a role not only in mammals but the other animals also. So that is all for today. If anyone is having any question, you can ask me. Any doubt is there or not? Yes, Anjali, you can tell.
Ma'am, I wanted to ask why germline stem cells, we can't call germline stem cells as totipotent. Ma'am, it
Germline.
was
Huh.
germline stem cells, yes. Why they can be called as totipotent? Ma'am, because,
Why they? Huh.
yes,
Yes.
ma'am. Yes, ma'am, listening.
What do you understand by totipotent cells? First, tell me
Um,
that.
Ma'am, the cell that can,
Hmm.
मैम
No, tell
जो
it.
पूरे ऑर्गन को मैम द सेल विच कैन जनरेट द होल ऑर्गेनिज्म or the
Uh,
any part of it.
yes, entire complete organism. Like
Ma'am,
it can,
yes, ma'am.
it can organism which can create into entire organism. You understood? Entire organism means not only the cell types but the extra membranes also. That is the
Okay.
ectoderm, endoderm, mesoderm, whatever was present.
Hmm.
So germline stem cell can develop into different other kinds of cells, but can it provide that membranes, extra embryonic membranes?
No, ma'am. Ma'am, actually, I thought that the gametes are for the gametes ultimately formed from these germ cells, and ultimately the organism developed from those
No,
entire.
no. Gametes
Right.
are not from these cells. Germ cells are developed later. Gametes are not from germ cells. One minute, I will show you one. Uh, uh, yes. Can you see my slides? Mm. Hello.
Yes, ma'am, I'm listening.
Yeah, can you see my slide?
Yes, ma'am. We can see now.
So here you can see like uh, totipotent cell are the embryonic stem cell, which can form all the cells of the body and the extraembryonic membrane, like the oocyte sperm, which will actually developing into modula blastula and that. But it is what you are telling is the uh, what you told is germ line cells.
Yes, ma'am. Are they totipotent? Can we call them totipotent? That was my question.
Germ line uh, stem cell means that not you are talking about this hematopoietic stem cell, no. This sperm or ova only you are telling, no.
Ma'am, actually, it was a question from assignment two of
हाँ
ours, and
हाँ
it was asked which like we have to choose which statements are correct or incorrect. So it was written there. Germline stem cells are totipotent cells. So with the logic that germline stem cells give rise to gametes, and then
हाँ
they will then further the fertilization takes place and they develop into entire zygote and then the
हाँ हाँ
whole organism. So my answer was yes. Why? Actually, I was wrong, and the sentence was incorrect. But I wanted to know why. Like what then? If not, then what should we call them? The germline stem cells.
okay. The germline stem cell, I will see the question because to my knowledge also what you are telling is correct only because germline stem cell should be totipotent only because they will give rise to the. I thought you are talking about the hematopoietic stem cells, but no. If you are talking about that, it is not totipotent. But I will see once the germline stem cells are
Okay.
totipotent, and I will
Okay.
like germ cells are obviously totipotent in nature only because they are only developing into an entire organism. So I will check and I will once speak with sir
Okay.
also.
Okay, ma'am. Okay,
Uh,
ma'am. And please do tell me in the next lecture. I will try to join.
yeah, I will.
And ma'am, I have
Yeah,
another question. Ma'am, it was a question from assignment three, and it was just a simple question, and it the we have to choose the options, and the questions are eosinophils are, and
हाँ,
ma'am, there were several options, among which two options we thought this could be like having acidic granules, and
हाँ,
this was one option, and the other option was uh, having acidic granules, and which we pointed out, like we which ticked which we like thought it was the right answer, but it was wrong, and the correct option was having major basic protein. Ma'am, I wanted to ask
okay.
why this is incorrect, like having acidic granules.
No,
Eosinophils are having acidic granules, yes ma'am.
okay. This is week. What? Which week?
Assignment 3. Assignment 3, it was, ma'am, it was a simple question. Eosinophils are, and the options given were primary phagocytes, first option, second option having acidic granules, third option having multilobed nucleus, fourth having major basic protein. And ma'am, among these we chose the second option having acidic granules, and but it was wrong, and
It was a multi-choice question.
yes, ma'am, it was the MCQs. The assignments
You
we
chose
got.
only one option.
Yes, ma'am. Actually, no, ma'am. Multi-choice. We have to choose any one option,
Yes,
and
choose any one option or multi-choice option.
ma'am, we have to choose any one option.
Okay.
And yes, we chose the answer. Eosinophils are having acidic granules. I mean, we have eosinophils, acidophilic.
But
We applied that logic, but it was having major basic protein. Ma'am, what?
huh,
Why?
actually eosinophils are not acidic in nature. No, eosinophils are containing basic proteins for which it is reacting with the acidic dyes. So it is
Oh,
staining that.
oh, oh, oh.
That are like whenever some basic protein is present, it will react with the acidic dye only. No, they
Yes,
are
ma'am.
known as acidophils, but
Yes, ma'am.
they
Okay.
are
Yes.
not containing acidic proteins.
Okay. Okay, ma'am. Uh, I think I have. And ma'am, ma'am, another question was it was a question of assignment week 4, and the question was which is not a function of cytokines. And
Okay,
ma'am, we chose the option cell death inhibition.
okay.
Ma'am, not a function of cytokine, but
Uh,
cell death inhibition.
can you just mention in the message like what are the assignments and what are the question numbers because that will be easy for me to check. because I have to check the answers as
Okay.
well.
Okay, ma'am. It was ma'am. Shall I speak or write in the
No,
comment?
in the comment box you write assignment this and question number this.
Ma'am, actually, it was ma'am. Actually, cytokines induce cell death, na?
Yes, yes.
मैम देन एग्जैक्टली सेल डेथ इनिबिशन तो ये करता नहीं है देन वाई इट केम रॉन्ग एंड द करेक्ट ऑप्शन वाज ऑक्सीजन ट्रांसपोर्ट बट वी हैव व्हेन आई वाज गोइंग थ्रू द presentations or the lecture materials it was written that it helps in oxygen transport it like increase the oxygen transport at the site but ma'am
Okay.
and it induces cell inhibition and the question was not a function of cytokine not a function ma'am how it was wrong oxygen transport.
Like no, no, you are telling like it was uh, telling like it is inhibiting the cell death.
Yes ma'am. The option was given cell death inhibition
Huh,
and from my knowledge cytokines induce cell death.
huh.
Yes ma'am, so how this became wrong.
So you chose this option like inhibition one.
Yes, ma'am. I chose cell death inhibition because apparently we have learned that cytokine induces cell death, but does not inhibit. And at the site, it increases oxygen transport. Ma'am,
Okay,
via through several factors, not the cytokine itself, through through many other processes or mediator factors, but the correct option was oxygen transport. Like it is not a function of cytokine oxygen transport, but ma'am, then it is cell death inhibition itself was not the its function.
okay. I have to actually see the questions because currently it is not in front of me. So I have to check the questions and the answers also whether it is wrong or not. So what you can do is you can just mention all the three questions which you told today, like not
Okay,
only
ma'am.
this.
Okay, ma'am. I will. Okay, ma'am. That was.
Week 4 question 2 then.
Ma'am, wait. That word germline stem cells I have to write.
हाँ.
Week two question. Ma'am, I guess I have one more doubt regarding assignments. Ma'am, can we take fungi as our microbiome in human body? Fungus?
Fungi.
Yes, ma'am.
Uh, Fungi is not that much present in our body, but what was the question?
Wait, ma'am, I will write and I will ask you as well. It's week 5, question 8. Okay, so we have to choose which statements are correct or incorrect.
Huh.
So first was like microbes are 10 times more than the number of human cells. It was correct. It was given in the PDF as well. And
Hmm.
second was an adult human swallow and digest about 1 liter of mucus. It
Hmm.
was again given in the PDF. And third one, it was there like it is there in the PDF that yes we fungi are like they can be considered as pathogen, but the in the question it's written microbiome present in the human body doesn't include fungi. Ma'am,
Okay.
fungi
Fungi are there, but very much less compared to the other bacteria. some viruses. So you have chose this answer.
Ma'am, actually are not correct. So, ma'am, according to us, it was correct that our fungi doesn't include in our microbiome, and it was the option. But, uh, ma'am, actually, it's wrong. Ma'am, it says the question says human body microbiome doesn't include fungi.
Yes, but
Yes,
that is
ma'am.
wrong actually. It includes fungi, but not to that extent.
Yes, ma'am. And actually, we chose the answer that it is incorrect. But again, the
It is wrong. Ha, understood. I understood. Okay, that
yes,
is
ma'am.
week 5, question 8.
Yes, ma'am. Week five, question eight. Yes, ma'am.
Okay, okay.
I mean, yes, I
These,
have these doubts. Yes, ma'am. I have four questions till now in the assignment. So I thought.
another question you write the number 3 you have written. Another one. I will first check the questions,
And
then
that you
check
tell that you told already.
that
The
eosinophil,
eosinophils
basic
one.
protein.
Yes, ma'am. Yes, ma'am. Yes, ma'am.
Then you will.
That cleared. I got that got cleared.
Okay, these three I will check and I will speak to sir if I find that the answers are wrong. And then if it is wrong, then we will again recheck the assignments.
Okay. Okay, ma'am. Okay.
Then next class I will tell you.
Okay, ma'am. Okay.
Okay.
Okay. Thank you, ma'am.
हाँ Okay, thank you everyone. Then I am. You can